Yeah.
Okay, good.
And I think it's 9 o'clock now. Dr.
Okay.
D, are you there? So, okay, Dr. Tran, do you want me to introduce a little bit yourself, or you can go ahead and introduce yourself?
Yeah, I can introduce myself. Yeah,
you can do it by yourself, right?
Yes, still many students want to join in, so maybe we could wait for a little time.
I see. Should we start, or this is the 1 minute?
Okay, I think we can start.
Okay, go ahead.
Yeah, because my student will be the TA, they can also admit student and also answer the questions.
Awesome.
Okay, so good morning, everyone. Welcome to our seminar series. I think right now you can see my presentations. So I'm Charlton, so the assistant professor in the Department of Biology at Harvard University. I'm bioinformatician. So today, I want to talk about how we use R in the biology studies. Especially we will show some examples how we use the R in the biostatics side of bioinformatics. Okay, so in today's talk, I will first introduce why we use bioprogramming, especially R in the biology, because right now biology is also a big data science field, as we have many kind of data to deal with. Then the second, I will introduce some basic knowledge about R. And then I will talk some examples, like how we use the t-test to do some analysis in R, and how we use R for the GWAS analysis to identify some mutation at genes associated with disease. So let's first talk about the why we should use R in the biology. I think nowadays maybe many people heard about the DNA, the genomes. So this picture shows the human genomes. So we have about 23 pairs of chromosomes. 22 ultrasounds, then we have 23rd pair determined either the girls or the boys. For this 23 chromosome, one from the mother and one from the father. When we talk about human genome, that means the genetic informations encode the DNA sequence within all these 23 pairs chromosome and also the mitochondrial DNAs. Our human genome include a lot of information. One human genome we have 3 billion base pairs nucleic acid. And I think everyone like read the Harvard box. A set of Harvard box is include total 1 million words. So if one word has average 7 characters, then about 7 million characters. If we think one base pair nuclei as 1 character, then one human genome will include information as many as 428 sets of the Harvard box. So if we put them together, that is the height of almost 40 to 50 story buildings, almost the height of the Washington Monument. To determine the information of our human genome, we call the Human Genome Project. This is the international project collaborated by about 7 countries. And this project actually started as 1990. We start sequence some small organisms like the E. coli and the rat mice. Then we learned some experiences about in the 2003. So we complete our human genome project. We sequenced about 99%. That total took about 30 years. And the entire project, it spent more than $3 billion. Of course, this is the first human genome. It's $3 billion, we cannot sequence many people's. But after that, the price, it dropped down. So in this picture, we can see the white line is Moore's law. That represents the normal speed of technology development. Then the green line, that is the cost per genome. We can see it reduced. But in 2007, when we sequenced Dr. Craig Winter genome, it still took about 4 years at $100 million. But after that, technology we call the next generation sequencing was introduced to the market. Then the next year to sequence Dr. Watson genome, it only took 4.5 months at $1 million. But nowadays, use some latest technology like Illumina X10, we can even sequence our genome in a couple of hours, and the price could drop to the $1,000. That means with the development of this technology, we can sequence our genome in the population levels. This is very good news, but it also brings some problems. For example, the left picture shows the growth of DB-SNP. DB-SNP is a database that include many kind of human genetic variations. We can see when the next generation sequencing in the market, the submission of the DB-SNP are significantly increased. So right now, the DB-SNP already include over 100 million of the genetic variations. But in our human, most of these variations, they are function neutral. So we want to find some that genetic variation play very important roles, like maybe associated with certain disease like cancers. So this is like find a needle in a haystack. So that is why we want to apply some computational program to announce this data. In the biology lab, usually we classify as general weight lab as a dry lab. So in the weight lab, we try to sequence our genomes, try to do the function study in the animals. In here, like how we use PCR, this is the basic technology we should have. But in the dry lab, like in the bioinformatics or biostatistics, most time we do not deal with these chemical solutions. We deal with the computational programming. Like today, the R we want to introduce today, and the traditional C++, and also the Python. I think this is a very popular language right now we use for the machine learning. So why we want to use R? I think in the biostatic analysis, statics, traditionally they use some tools like the SPS and SAS. So I think we look at the Herbert movies. So the SAS users, they are very like the mongoose. So they are very limited. They rely on the this algorithm or function developed for them. So when we have these new functions, we must wait for the SAS, the program, they build the function in the software, then we can use that. But the R users like the wizard. So they can rely on these functions, but they can also create their new function use R programming. Especially many statics, they develop some packaging in the R's. once the package in the online, we can download them and install this package and use that. So for the R, right now there are over 10,000 R package. So that means when the scientist develop this R package, put it on the website, we can directly download that and without any kind of delays. Not like the SPS or SAS, we must wait for their scientists build the function in the software. And also very important is sometimes we can customize this kind of the package. For example, today we are introduced, we can install some package like QQman. Then we can modify these functions and calculate the p-values and draw some pictures we want. There are many kinds of the R package for the biology analysis. For example, this bioconduct. So this bioconduct they provide to us for analysis high throughput genomic data, like we just taught us that how is next generation sequencing to sequence the genomes. The bioconduct they use some R study programming. It's open source and it continues to add many kinds of the package used for the genetics and genome analysis. So currently there are over the 1,700 software package there, and you can search that, search for the appropriate package used for your own studies. Okay, in the second part, I will talk about how we what is the R. So the R is the free open source package, so everyone can download that. It's developed earlier by the BIOS lab. So this actually is a very powerful package. Many biologists use it for the static analysis, but actually it's also the programming language that we can use to write some script. It crosses the platforms. That means you can develop in the Windows, but then you can also use it for the Linux and Mac. It also has an interface for the other language like C++, Python, Perl. That means you can combine with R with this language and build some pipelines. I think very important is use R, we can manipulate the array and matrix. In many times, like the microarray experiment, the data they are present as matrix, so we can use R to deal with that. The R is already has some built-in static functions, and also the latest R functions, they are developed by the R. We can install that and use that. The R, they has many kind of state-of-the-art graph captures, like the plot or QQ plot. To use this function, we can generate very high quality data. Especially this picture are very good for present the genome data. Like we introduced before, the R, they already have many kind of the package used for the biostatistics and bioinformatics analysis. So how we get the R? So most time we are downloaded from this comprehensive R archive network, CRAN. There has a free R package there. It has a Windows versions, the Linux and Mac versions. Some students, they like use R Studio, so basically it's very similar. So if you do not install the R in your own computer, I think you can also directly run R script in this web browser, rdr.io. But I think I strongly encourage you to install the R package in your own computers, because later you will use that for your own data analysis. Okay, this is the R graphic user interface. So when we open our R software, we can usually see the R console. We can also open the R editor. To run R script, we have two ways. One is we can directly type in this R console. Or maybe we can put our script in this R editor. The advantage is that we can save this script, and then we can reopen it again in the future if we want to use that again. So we will show later in our R lab. Let's review some basic ideas in the R. So in the R, so actually we have this idea is we call the object. It's like the variable in the other language. So object is that way is we collect the atomic variables and also other objects and put them together. For example, when we have the microarray experiment, we have the problem intensity. We also have the collect data from the patient data, like some tissues, different tissue diagnose, follow up. Then we have our gene data, like sequence ID, annotations. These variables usually we put in some matrix. Atom data, we include some numbers. For example, integer, double, complex. We also have some characters, logic, and the function type. The object has three forms. The vectors that is we usually collect the data of the same data type. Then the matrix. So matrix, I think this is the most general data format in the R. So it's a rectangle table of data of same type. And then array, actually this is a high dimensional matrix. Let's show some examples. For example, we know the vectors, that is the order of collection data of the same type. In here, for example, in this object 1, we include 2479. We can assign to some object, then we can manipulate this object. For example, we can times 2, then each number in this object will be double. One example, in the microarray experiment, for example, the mean spot intensity of all 15,000 spots on a chip, this is kind of the vectors. So in R, a single number, this is a special case of vector with one element. Other vector maybe include some character strings, for example, our sequence data or some logic vectors. The matrix is a rectangle table of data of the same type. This is most common in the R. For example, when we have the microarray experiment that include an expression value for 10,000 genes for the 30 tissues. So this is a matrix. We have 1,000 rows. Each row is a gene and 30 columns. Then each column is a tissues. Later today, we will learn how we generate a matrix use this matrix function. For example, use this matrix function, we can generate a matrix with 2 rows and 3 columns. Matrix actually is two-dimensional, but if in the 3, 4, or higher dimensional, that is we call the arrays. For example, in here we show the red at the green, foreground at background for two sunspot on the 120 chips. This is 4 times 20,000 times 120. This actually is a 3-dimensional array. Okay, this is basic for the R's. Then let's talk about the application for the how we use R in the biostatics. So we know statics is a way how we learn from the data. Actually, this is very important when we do our research. Study we should consider all elements when we do the study designs, data collection, and do the data analysis. We also need the judgment. For example, we generate the p-values. Biostudy actually, this is a study we apply to the biology and the health problems. For example, when we do some experiment, then we should think about our study designs. Okay, what are you trying to learn? For example, you want to do some case control association study and identify the genes associated with the major depressions. How you could approve this. Then we will think about sample collections. Who are you going to study? So you want to collect some patient with major depression and also health peoples. How many people you want to collect? And then we should think about what should they be collected. You should collect their blood sample for DNA extraction, and you want to collect other kind of phenotype like their blood pressures, height, weight. Then you want to do some data analysis. You want to think if there are any kind of effect. In other words, you want to find some association between some genetic factors with the disease. And finally, do the conclusion. What does this mean? Actually, all this is kind of the biostatic and also bioinformatic questions. What kind of the test, like Fisher exact test, you want to apply to this kind of study? You want to do some power analysis and try to determine how many kind of samples we want to collect. So the R actually it provides many kinds of the package for the static analysis. This could come from the different resource, different authors. And many kinds of study research, they develop some new method, and this method is provided as R package. So use R, we can install this package and then apply this latest method to our studies. So here is just to show some very simple example shows describe studies. For example, when we have a weight like data, we include many numbers, then we can calculate their mean is mean functions. We can also use like SD, this function calculates standard deviation. When we have many numbers, we can use mean and max function to find the minimum as maximum variables. We can use quantitative calculate the 25 at 75% quantities. And the summary function basically could return the many basic statistics like mean, medium, mean, max. This is just a very simple function, but actually the R also provide many advanced functions for data analysis. I think next week we will have a workshop introduce regression models. A regression model could include linear, nonlinear, but it will also has a R package provide function for the time series analysis, like clustering. That is we can use it for the microarray experiment. Another advantage for the R is R actually it provides many kind of functions like the plot function. We can draw very high quality pictures. For example, today we can draw two pictures like we can box plot, we can compare two groups at the Manhattan plot. We can try to identify some target. The R provides this function and you can change many arguments and draw high quality pictures that is used for your publications. Okay, today so I want to show one example for the cheese test. When we talk about the studies, I think very important idea is the p-values, the hypothesis test. So hypothesis test basically we want to know if there are any kind of difference between a group, or maybe we want to judge if this difference is just random by chance. So here we will test this probability that is we call the p-values. But here I want to emphasize p-value actually is not indicative of the size or importance of the effect. So no significant do not mean no effect. So in our study hypothesis, basically we had two types of study hypotheses. One is we call the null hypothesis. That means usually no difference between the means. That is why we compare the two of the different group. Or maybe it also means the correlation is zero in the population. That means no associations. Then in the opposite alternative hypothesis, that means we do have the different means and we have some correlation in the populations. We could calculate the p-values and then we can set up some cutoff signaling levels. Usually we do it 0.05 or the 0.01. So if the p-value is greater than this 0.05, then we do not reject our null hypothesis. That means no difference. So most time we want to find the p-value is smaller than 0.05. That means we could reject this null hypothesis. In another word, it also supports this alternative hypothesis. So because when we do some comparison, many times we want to see this difference between the groups. Okay, so today's we'll talk about the t-test. The t-test is based on assumptions that data come from the normal distributions. For example, here is shows an example we can have this daily energy intake from the 10 peoples. Then we want to investigate why with this energy intake is system different from our recommended value of 6,000 kg. So basically, that is we want to test if there are any difference between the mean value of our sample group. We want to compare with the population levels. So we can assume this data come from a normal distribution. Then our object is try to test whether this distribution has these mean values. So we can use t-test to do that. I think two sample t-test is more combo. For example, when I want to compare two groups, like the patient and health peoples, the two sample t-test, we can test hypothesis that these two samples we usually assume to come from the distribution. Then we can test whether we have we can calculate a p-value to think if we have a difference between these two groups. For example, in here we have the two group, obviously at the lean group. We can use the t-test test with there is a shift level between these two groups. So in the R, of course, there are many kinds of functions. When you try to have some new function, you want to use the help functions. Then you put your t-test there, then it will into detail of these functions, what kind of the arguments there. Then you can modify that. You can want to do the two-side or one-side test. You want to do the paired t-test. You want to modify the confidence levels. Okay, so the last part is that I want to show the how we use R for in the bioinformatics. Okay, so we want to show some example how we use R for the Jiva study. So let's talk some basic idea for the Jiva side of bioinformatics. So when we bioinformatics, you can look at this word, is bio and informatics, right? So when we do our biology study, we research the genes, proteins. But in our humans, for example, in our human genomes, there are more than 20,000 coding genes there. So we must research these genes in the systematic levels. That means we should do research in the genomes or proteomics. So because we have so many data, that is that we must use computation tools to analyze this data. We must store this gene or protein information in some database, and we need to develop some algorithm and also static models try to analyze this data. I think a central question for the biology is we try to figure out the relationship between the genotype and phenotype. For example, in our genes, we have the sequence variant. This is the DNA difference in the populations, right? We want to identify some sequence variant. For example, this sequence variant may be carried by some patient. We want to find some sequence variant associated with certain disease, like the major depressions. I think nowadays, maybe for the COVID-19, you may heard about this mutant we call the D614G, okay? So basically, we want to try to identify this kind of mutations that may be associated with COVID-19. Sequence variant are actually very important. So when we look at this familiar face, we can know, okay, this is Will Smith, this is Jack Chan, okay. We know that everyone is different because everyone we carry the different sequence variant. In sequence variant, we can research many kinds of biology questions. For example, we can know how our ancestors we move from the African and then how they move to the other part of the world. And also, very importantly, when we sequence the patient, then we can learn which sequence variant may determine the disease. So how we do that? Usually we collect the mutation, the DNAs from the patient. Like we can either do in the population level, we can collect some of the patients and some of the health people, or sometimes we can collect the patient and also health people from the same family. Then we can use next-generation sequencing to sequence their DNA to try to get their genome information. Then we compare with the reference genomes that determine the sequence variant. But usually, For the genome, we will have hundreds or thousands or even millions of the sequence variants there. Which variant is the most important? Which variant could determine the disease? Then we need to apply our biostatistics and also bioinformatics approach. We try to determine the most important target. When we identify this target, we can do some function study, like do some knockdown mice. If we validate this real targets, then we can think about how we design the drug. Okay, this is we also call the precision medicine or personalized medicines. The idea for do this case control assertion study is very simple. Okay, so what we want to do is we will try to identify the variant that only present in the case in the patient, or maybe they are enriched in the patient, but just present a few health controls. This is we call the risk variant. And also in other side, we also want to identify some variant only present in health peoples, or maybe they are enriched in health people. This variant we call the protective variant, but they are also very important because they can be the target for the drug designs. So in this process, we should apply some association tests to identify this variant. For test this association, we use this genome-wide association study we call the GWAS. This test is usually we apply some next-generation sequencing or SNP array. We can screen the markers in the DNA's genome of many peoples, thousand peoples. Then we try to identify this genetic association with a particular disease. When we identify this target, then the research can use the information, try to develop some bait strategy to detect and prevent the disease. So GWAS is a very powerful method. Many GWAS studies have been used to identify variant associated with common disease like the cancers, diabetes, and also psychiatric disorders. GWAS usually include the 5 steps. Actually, every step we will apply, use the biostatic and bioinformatic approach. For example, we try to select the association measures. Then we need to select what kind of test for this association. For small sample, we can use the Fisher exact test. Large sample, we can think about the chi-square test. Then today I just show 2 examples how we use R to visualize and interpret our data. When we have the data, we also need to consider as multi-test correction like buffering. And then the last, we should use some independent sample to do some replication strategy. For example, we can use R to draw some QQ plot. QQ plot, this is the picture we can see the observed p-value from the null hypothesis. So we get the p-value for each SNP, then we can sort from the largest to the smallest. Then p-value, because we use log p-values, so we can that means we sort from the smallest to the largest. For this QQ plot, for example, in the picture B, we can see some diversion in the tails. So that's good. We see some kind of, here means some SNP, they have strong association with certain disease. But at the beginning, they are always follow the expected p-values. Then in the picture C, if we draw the QQ plot like this, we can see their earlier diversion, that means we may have some experiment design problems. So the QQ plot we can also use for the quality control in our biology study. So Manhattan plot actually is the most important features in the GWAS study. So here, this picture shows the Manhattan plot. Each dot means each SNP, each sequence variant. So we calculate the p-value of each sequence variant. Then this p-value are represented by the genome positions from the 1 to the chromosome X. Then this y-axis is their log p-values. If we have very significant association, that means p-value are very small, then their no p-value is very big. So we can send some cutoff. Here we can see there can be some peak in the chromosome 3. So this region could be our target region. So the variant or gene in this region may be associated with certain disease. Okay, I think this is basically the presentation we covered. So later I will show some the handout and send you yesterday, and we will do some practice. Okay, so before our lab, do you have any questions?
Hi, this is Nani from UTC. Thank you, Dr. Stan, for your very nice presentation. I think this is really good for our student audience. There's our tutorial, and especially you talk about the R-base, because before my topic next week. I think this is really useful for those students, especially they have never touched R. Thank you. And I do have a question about the GWAS analysis. So I did some research when I was working for St. Jude Children's Research Hospital. Many I do research on research on identifying differentially gene and gene set. So yeah, I just wondering when nowadays are you still working on microwave-based gene expression data, or you do data analysis on the NGS, next generation sequencing data analysis?
Yeah, I think the recently I think more and more people, they try to use the next generation sequence data, because advantage is the traditional GWAS, we only look for the common variant.
Right.
In next generation sequencing, we can identify use this common variant, we do the analysis like GWAS, but we can also use next generation sequence data to look at the real variant and also some large variant like copy number variations. So this is the huge advantage for the next generation sequencing when we do this association test.
All right. Yeah, it looks like that's the NGS data is more popular than the traditional microwave expression data. Thank you.
Thank you. Okay, so I will start the our lab. Okay, so let me share my okay, so here actually this is shows the this is an OpenMyR software. You, of course, if you didn't install that, you can direct type your script in the web browser. For example, we could run our R script like in the console. Like for example, we can do some very simple calculations. Okay, like 2 + 1 = 3. But we can also try to open some like a new script. We can put our script there, and we can save that, and later we can reopen that. To save time, I already put my script in the R editor. Okay, then we can directly run that. So in the R, we have this number signs. Oh, this is a comment. Okay, so actually, our software do not exclude that. First is we try to want to correct some object. For example, in here, and try to correct the object 1. Object 1 equals C2479. Okay, this is our object. Okay, we can run this script. We can use the Ctrl R. We can run this script in R console. You can see this object. Okay, then we can value our object. It will include 4 numbers. We can do some manipulations, like for example, we can object times 2. Okay, we can see actually every numbers they are doubled. This is object. Okay, then in this object actually we could correct some matrix. So in here, we can use these matrix functions. Okay, matrix function actually we have our vector of data. You have a real data. Okay, then you should space how many rows and how many columns. For example, in this matrix function, we have this 1, 2, 3, 4, 3, 4. Okay, this weight has 6 numbers. Then we want to have 2 rows and 3 columns. Okay, then we can run this script. Okay, we can look at this matrix. Okay, so for example, this matrix will have 2 rows and 3 columns. And then, if we want to look at the first rows, we have this M1, we have this brackets, then the first number will indicate the row numbers. Okay, second number will indicate the column numbers. Because we only want to look at first row, you can put the 1 there, but leave the column numbers as empty. When you run that, okay, you can see in this M1 we have first row 1, 3, 3. Or sometimes you want to look at the second columns, then you are left the first number is empty, then put the 2 in the second options. When you run that, we can see this is actually the second column, 3, 4. And sometimes you want to look at some numbers, for example, some number in the cell of the first row and the second columns. You want to have this space row and the column numbers 1 and 2 there. So this is 3. Okay, this is the the number in the first or second numbers. Okay, this is just a very basic ideas how you assign some variable in the R. Okay, so then we talk about second part about this very simple biostatistics test. For example, in here, I'm trying to use this R normal function. We can generate about 1,000 numbers. Okay, then we look at our X1. Okay, so here we have 1,000 numbers. Okay, we have many numbers there. I think if you want to calculate the mean values that by hand, it takes some time, right? So use R, very simple, we can use these mean functions. Okay, we can mean functions and we put our object there. Okay, we can get the mean values here. This is the minus 0.005. Okay, very close 0. Then we can use this SD function. We can calculate the standard deviations. Standard deviation is about the close 1, 0.990. We can also calculate the quantities. I think in study test, main time we use 25th or 75th percentile. Okay, then you run this function, you quantize, then you the vector you put the 75th, 25 or 75 percentile. Actually, you can change these numbers. Okay, you want to look at the 10th or 90th percentile sometimes. Okay, then you get the 25th is minus 0.7 at the 75th is 0.69. Then because we have 1,000 numbers, okay, it's very difficult to find the minimum and maximum value by eyes. But actually, you can use mean functions. Okay, you put your weight there, you can determine. Okay, the minimum value in this 1,000 number is minus 2.9. Then you can use the max function to determine the maximum values. For example, here the maximum value is about the 3.12. Okay. Sometimes we want to have one function that is tell us the basic statistics for our R test. We can use these summary functions. Okay, we can run the summary and the X1. Actually, here we tell several important values. For example, the mean values. Okay, the first quantile, 25 percentile. That is what's the medium. Okay, then tell us the means. Then here it shows the third quantile, and also we give the maximum values. Okay, this one function we can get the basic describe statistics for our data. Okay, so then we will tell a little about the p-values. Okay, about the cheat test. For example, we introduce the one sample test, right? We have a small group. We have 10 people. Then we get their daily energy intake. Okay, so we want to put these values from 5,000 to 8,000. We want to assign to a variable this object to an object. Okay, this object you can name kind of any name, but you should. It's better you have a very meaningful name. For example, here I name as energy tag. Okay, so we have 10 values. We can look at this energy tag. Okay, we have this 10 values from 5,000 to 8,000. Okay, then we can first we can calculate the mean values. The mean value is about 6,496. Okay, in here we have some recommended values. Recommended value is 7,600. Okay, it looks like the difference, but how we should calculate the p-value to judge. Okay, we tend to think there are some difference between these mean values with our the recommended values in the populations. Okay, we will apply the t-test. Okay, we will have our t-test here. Then we will have our energy tag. Put our object there. Then we have our recommended value. The mean value is 7,600. We could run that. This is one sample t-test. Okay, we have the t-test values, degree freedoms. Okay, I think the most important is we try to determine the p-values. Here, p-value is 0.008. Okay, it's much smaller than the 0.05 or even smaller than 0.01. Okay, this means there's some difference between our samples with this recommended p-value or with this recommended energy tag. They also give other very important, like for example, the 95% confidence intervals. Okay, and also it shows the mean value of our small samples. Okay, this is once the t-test can tell us. Okay, so then the next is this one sample test. Okay, we compare the means in our own sample with the population. Then meantime, we try to compare two groups. That includes a non-pair group and pair group. Okay, here we try to look at the non-pair group. In here, we need to install a package ISWR. Okay, so if you first time install the R in your computers, then you need to install the package with the ISWR. Okay, and if you install that, you can run your libraries. Okay, library ISWR, that means you load this package to your workspace. You can use that. Okay, otherwise you cannot use some kind of the energy data set. Okay. Okay, I have this library, then I can attach energies. Okay, I can look at the energy. Okay, we can look at energy. We have the two columns, right? The first column expands on values, then the second column is status. That is actually include two groups, okay? OC group and link group, okay? What we want to do is we want to see if there are any shift between these two groups, okay? So that means we have this values for OC group if there are any difference with the link group. We can use this t-test to do that. We have t-test, then we have explain these values, then stages, this is the group. We could run that. Okay. Here, this two sample t-test, we have the t-value degree freedom, then we look at the p-values. P-value is here is 0.0014. Okay, that is means this is a support alternative group. Okay, so this true difference is not equal 0. There are some difference between these two groups. We can also get their 95% confidence interval. Okay, we can look at their means. Okay, like in the link groups, the means is 8.0, and then the opposite group is 10.29. Okay, it's a look at the unitake opposite group is greater than the link group. But here we can use p-value to judge that. So this is for the values, but you can also run the box plot. Okay, when you run the box plot, okay, you can get very clear the value in the link group is smaller than the opposite group. Okay, when you publish papers, you can get the p-values, and then you can also show this box plot. Of course, today I'll introduce very simple study test. I think next week Dr. Gao will tell about more advanced analysis like a regression test. Okay. Okay, the last example I will show some Java study. For example, here we will show the QQman. Of course, if you didn't install this package, you should use install package QQman to do that. Here I already installed that, then I can load this package. Okay, library QQman. Okay, we have the Java data. Okay, So I use head function to only show okay, so let's see. Okay, so I think I have some problem for the QQman. Okay, I need to install this package again. So, for example, I can also use this function to install the package. We can scroll down. Okay, we have QQman. We try to let's install this package again. Okay, if I think load this QQman. I don't know, in this computer, it's not wrong. Okay, so let me try to use the web browser that it also can work. Okay.
Hey, Dr. Tang, I'm
Yeah.
getting, I think you need to install RTools, the new version of RTools, in order to use QQman in
I
the,
see. Yeah.
on the 4.0. So.
Yeah. So. Okay, so I think for showing purpose today, I can just run in our web browser. Okay, so we can run this script. First is that I want to load the QQman. Okay, then we can look at our GWAS result, and we can draw the Manhattan plot and draw the QQ plot. Okay, use Manhattan and the QQ functions. We could run that. Okay, so here, this is our result. Okay, so basically, this GWAS result actually includes more than 10,000 SNPs there. Okay, we just show the part of that. We have our SNP, then we have their chromosome. At the base pair, they said these are their positions, and we have their p-values. Okay. What we want to do is we want to look at all these 10,000 p-values. We want to look at if draw some QQ plot to look at if they follow the expected p-values. Okay, so in here we can see at the beginning for expected p-value, but in tail there are some diversions. Okay, that means this kind of the SNP maybe has a strong association with certain phenotype like disease. Okay, here is shows the Manhattan plot. Okay, in this Manhattan plot, actually each dot is a SNP. Then we calculate their log p-values. We can sense there are some SNP in the sequence variant in the chromosome 3. They has very strong associations. Okay, actually this variant is here some SNP has some diversion in the QQ plot. Okay, when we identify the these regions, we can look at the variant at genes. Then we can use this variant has gene as target, we can do some function study for the drug designs. Okay. Okay, so of course this is just 1 hour workshop. We cannot cover everything. Okay, we just cover very basic in the R's. And next week, for this workshop, we will continue to talk about the regression models. Okay, so I hope the student can continue joining this workshop. So, okay, so then, do you have any questions?
Hello.
Hello.
Yes, I have a question, which is, is R gonna be capable in running distributed memory and like message passing, shared memory, those kind of things?
Could you repeat that again? So.
Did you hear me or?
Yeah, I can hear you.
Yes, is R
question?
capable of running sequential parallel processing, like shared memory, distributed memory? Like message passing?
I don't know. So do we have the feature in that? So you mean the memory management, right?
I bet about the Noma, I think he's worried about, he's concerned about the, it's like whether or not this software can be run on the parallel computing platform, like the shared memory.
Yeah, actually, we are in my project. Actually, I'm use this R functions in the for this HPC. I can write some script and import the R function there and send to the cluster. There are no problem for R. At R, they also have some functions that you can limit these memories.
I had a question.
Mm-hmm.
Good morning, Dr. Tang. I just wanted to ask, are there any particular functions in R that are good, particularly for analyzing secondary data?
What kind of secondary data you mean?
Secondary SNP data. So this data would have already been collected and we'll have, let's say, for example, the alleles like noted for each of the SNPs, let's say for about 50 SNPs. Any functions that you would recommend in R to analyze such data?
Yeah, I think this very space actually, the R is for example, today I show this Manhattan plot. I think in there they also has a certain test. It could calculate p-value for each SNP, and there are also some other like the SCAT. This function could calculate association for the real variant or even for the copy number variations. So there are many kinds of R patches there. I think based on your very space data, you can choose a different one. So maybe we need to search, yeah.
Okay, the Manhattan plot is a good one to start. All right, thank you.
Yeah, so okay. So any other questions? Okay, so if no questions, you can also send the question later. We can talk more detail. Yeah.
Dr. Tang, I'm sorry. One more question.
Uh-huh.
So I I noticed something happened earlier. I tried to download QQplot. I have R version 4.0.2, and it was saying that QQplot is not available for this version of R, and it's the most updated one. I'm not sure what's going on.
You mean QQman, right?
No, I was able to run QQman, but I tried to run or use QQplot, and it wouldn't load.
Oh, the QQplot. I think you need to also install the QQplot package. Then you can run the QQ.
Yeah, so I tried to install the package. That's what I'm saying. I tried to install the package, but I was getting an error message that QQplot is not available for the version of R that I'm using. And I just downloaded R, so it's the most up-to-date version.
Oh,
so I'm not sure why that's happening.
Yeah, probably the reason is this QQman is quite old, so they didn't update that.
Okay.
Yeah. But I think if you can send me email, so I have some link like tell you how. I think you most focus on genetic study. I have some links they teach you how to do the genetics, how to draw this different plot, how to do some associated test.
Okay.
Yeah.
Thank you.
I can send that to you. Okay. Yeah.
Oh, yeah. Before we stop, just the next week, Dr. Gao will keep on. I think continue talking about the. I'm sure that's R, right? Is R programming, and the first thing next week at same time, maybe it's the same time, and the second thing is that I'm going to upload this is the seminar in the YouTube. Please encourage your student who did not show up today, and to take a look at, and then do the survey. Submit the survey. I'm going to send the survey to you guys as well, so that encourage the students and even the faculties to submit this survey, because NSF, they do need it. Yeah.
Okay.
Yeah, and Dr. Turn did a very great job. We enjoy it. Yes.
Okay, thank you.
Yeah.
Thank you so much.
Yeah.
Thank you. Very good.
Thank you, Dr.
Thank you. Okay.
Thank you.
Yeah. Okay, we'll see you next week.
See you.
See you there.
Good job.
Bye.